Hello, my name is Emily Pace, and I am a senior at the University of North Georgia in Dahlonega. I have been working on research with the UNG Biology Department on the effects of ocean acidification on sea urchin larval survivorship and development. If you look at Figure 1, you can see the chemical processes of ocean acidification. The higher concentration of CO2 in the atmosphere causes an increase in CO2 being absorbed by the ocean. A chemical reaction occurs that increases free hydrogen ions, bicarbonate ions, and carbonic acid. This reaction also decreases carbonate ions in the ocean. Carbonate ions are important for organisms that use calcium carbonate for their shell and skeleton. Sea urchins are one of the marine organisms that have a calcium carbonate skeleton and may be impacted by ocean acidification. Sea urchins are important in many marine food webs and are important economically because they are used in important commercial fisheries. One previous study was done that looked at pH on multiple sea urchin species and found that acidic environments delayed developmental timing. We decided to look at a local sea urchin species, Lidocynus variegatus, that is located in the Gulf of Mexico and the areas surrounding the U.S. southeast Atlantic coast, and we subjected this species to low pH to look at survivorship and developmental abnormalities. We purchased green sea urchins from the Gulf Specimens Marine Lab. In a lab setting, we induced spawning by injecting them with potassium chloride, which caused sea urchins to release their gametes. After spawning, we added the larvae into their respective pH groups. We looked at the pH groups 8.44, which is our control, 8.0, and 7.60. We decided to use 8.44 as our control because of the pH of the water the urchins came in. Our treatment groups were decided based on projected ocean pH over the next 100 years. Every 24 hours, we looked at survivorship and developmental abnormalities. For developmental abnormalities, we took 10 milliliter samples and fixed them with glutaraldehyde. At a later date, we took these samples and looked at phenotypic differences between treatment groups. For survivorship, we took 4 1 ml samples for each replicate and looked at the ratio of living to dead larvae. Looking at Figure 2, it shows representative morphologies of 4-armed Pluteus larvae at day 8. The pictures shown are the most common phenotypic differences found between treatment groups and control. Picture A shows a larvae from the control treatment. Picture B represents missing limbs. Picture C represents yeeting of arms. Picture D represents unequal anterolateral arms. And picture E represents A pointed at a oral side. As seawater pH decreased, developmental abnormalities increased. Figure 3 represents the survivorship of larvae reared at varying pH levels. The y-axis represents survivorship in percentages, and the x-axis represents the amount of time the larvae were in their treatment groups. We looked at every 24 hours. The control had 100% survivorship until day 5, and then started to decrease in survivorship. For a pH of 8.0 and 7.6, survivorship started decreasing at day 5. Larval survivorship was significantly lower in pHs of 8.0 and 7.6 when compared to the control 8.44. Figure 4 shows a total normal versus abnormal percentage for phenotypes in larvae reared at varying pHs for day 8 of the experiment. The y-axis is percentage, and the x-axis is normal versus abnormal, and each pair of bar graphs represents a pH. As pH increased, abnormalities in phenotypes increased. Figure 5 represents the specific phenotypic differences in the larvae reared at varying pHs at day 8. We quantified 200 sea urchins per treatment and counted the amount of larvae that had unequal anterolateral arms, missing post-oral arms, yeeting of post-oral arms, curving of post-oral arms, pointed aboral side, and sharp-edged aboral side. As pH increased, phenotypes varied from the control. We can conclude that ocean acidification does affect survivorship and development in sea urchin species. Survivorship will decrease and developmental abnormalities will increase as the ocean acidifies. Why do we care? As the ocean continues to acidify, there will be adverse impacts on marine organisms that have calcium carbonate structures, including sea urchins. As stated previously, they are important to many food webs, for example, kelp forest, as well as to commercial fisheries. We are currently studying the effects that salinity has on larval survivorship and development, and for future studies we are going to look at the effects that lower pH and salinity have on sea urchin larval survivorship and development. Thank you for coming to my poster presentation. I hope you have a great day.